Hello guys, now we are starting our short notes video number 4 of microbiology. For previous videos, you can go to the playlist section of microbio. For any doubts, you can comment on our videos. So let's start. So our first point is complement fixation test. Okay, so this complement fixation test, it detects specific antibodies in serum, okay, by two stage system, okay. And this complement fixation test is used in, very commonly it is used in gonorrhea, okay. And next point for this complement fixation test is how to tell that it is positive or negative. If antigen, AG means antigen, if antigen binds with complement, okay, that means positive test, okay. If antigen is bind with complement, it is positive test, okay. But and how to check like the complement is bind with the antigen or not, okay, they are bind. So to check, what we do to check complement binding, like they are bind together or not, okay, we add sheep RBC red blood cell, okay, in that solution. And if hemolysis occur, that means complement did not bind, okay, so that means negative test, okay. See, if the antigen and complement are bind together, that means positive. But if they are not bind together and we add that sheep's red blood cell, so those red blood cells, okay, will be hemolyzed, okay, because of complement, because complement do not bind with antigen, so it will hemolyze the cell. So in this, usually the question is asked like this, like there is a hemolytic serum against sheep RBC, okay, hemolytic serum against sheep RBC and something like that, okay, they will form the question, okay. So this is negative and this is positive. Let's move to our next point. So that is our botulism, okay. And the agent that caused this botulism, agent is our clostridium botulism, okay. This causes flaccid paralysis. In this, actually what happens, there is no acetylcholine, and if there is no acetylcholine, so therefore it will cause respiratory arrest, okay. And we can also call, this is also known as no acetylcholine, this is also known as aka also known as neurotoxin shock action, okay. Now next point, it is usually present, okay, this is present in canned food, the food that you will eat in cans, okay, canned food, okay. It's of tennis racket shaped, okay, it is gram positive and spore forming, forming, spore forming. And the medium in which it grows, so the medium is kit, kit, tarossi medium, okay. And the test that we do for this is neutralization test we do for botulism, okay. In this test, actually, usually what happens, what we check the ability of antibody to neutralize antigen. AG means antigen, so ability of antibody to neutralize antigen, okay. Now our next point, see, if you want to check like the microbial count in water or air. Okay, so if you want to check the microbial count in water or air, okay, so in this we do like basically we count all bacteria. Okay, so all bacterias are taken into account or consideration, okay, account, which have grown in nutrient medium. Okay, so if you want to check the microbial count in water, okay, or air, so to check that we will count all the bacteria that grows. Okay, and if you want to check that the water is contaminated with sewage water, then we will do check that E. coli. Okay, in previous video we already discussed that part. Okay, now our next point is about Mycobacterium tuberculosis. Okay, so as we know, Mycobacterium tuberculosis have Cordy factor. Okay, this Cordy factor basically it does what? It inhibits leukocyte migration. It inhibits leukocyte migration. Okay, and this Cordy factor gives twisted ropes appearance as it sticks bacteria together. Okay, as it sticks bacteria together. Okay, and which test we do for Mycobacterium tuberculosis is that skin allergy test. What is that skin allergy test? Can anyone tell me? Skin allergy test. That is our Mantoux test. What is the test name? Mantoux test. Okay, skin allergy test. Mantoux test. In this test, actually, what we do, we will put in the skin dermis layer. Tuberculin is injected. Tuberculin is injected in the dermis layer. Okay, and in a normal person, if that increases by like the size of I think 20 mm, or if you will see a red rash, okay, then that means the reaction is happening. So it is our positive test. Otherwise, if the rash is not happening or it is not becoming wider, okay, so it is immunity is absent. Okay, so if it is negative Mantoux means there is no rash, then we have to give BCG vaccine to the patient. Okay, if this Mantoux test or tuberculin test, same thing. Okay, and if it is positive Mantoux, there is reaction happening against this tuberculin. So that means there is antibodies present in the person's body and if the person have already antibodies against tuberculosis or Mycobacterium tuberculosis, then we do not have to give vaccine. Okay, so immunity is already present, so we do not have to give vaccine. Immunity is present, no need to give vaccine. Okay, now some facts about this Mycobacterium tuberculosis. This is our rod-shaped bacteria. Okay, it will give pink color after staining. Okay, this is also known as aka Koch bacillus, Koch bacillus. Okay, and staining, it is stained by Zehel, Z-I-E-H-L, Zehel, N-E-I-L-S-O-N, Zehel-Nansen staining. Okay, it is stained by this staining and gives what color? Pink color. Okay, now our next point. In immunodeficient people, okay, these immunodeficient peoples lack what? They lack T cells. Okay, as you can correlate this thing with over that HIV virus. Okay, that HIV virus attacks on our T cells and causes immunodeficiency, that AIDS. Okay, human immunodeficiency virus, HIV, causes AIDS and it attacks on T cells. Okay, so you can relate with that thing. So in immunodeficient people, usually, so they lack T cells, because of which their immune system is weak. Rosettes, in this you will see rosettes with sheep RBC. Okay, RBC means red blood cell. Okay, rosette with RBC is seen in these people. Okay, now our next point. If in question they asked you like fast test, quick test, or express test, okay, or express test, that means they are asking for immunofluorescence test. Okay, fast test, quick test, or express, EXPRESS, express test. Okay, to detect anything, that means they are asking for immunofluorescence test. Okay, now our next point is about ELISA, enzyme linked immunosorbate assay. Okay, is used if antigen in blood is in low amount. Okay, and remember this ELISA is also used in hepatitis. Okay, in hepatitis. Now our next point, gram positive and gram negative is differentiated on the basis of cell wall. On the basis of cell wall. Okay, their cell wall structure and chemical composition, they differs. Okay, here only I want to tell you some bacteria that are gram positive. So there is a mnemonics to learn this thing. Okay, that is BADSS, BADS. Okay, so B for botulism, A for our anthrax, D for diphtheria, S for streptococcus. Okay, streptococcus, and another S is for staphylococcus. Okay, and for gram negative there is mnemonics get very, SHY, get very shy, like me. I'm just talking. Get very shy. G for gonorrhea, V for vibrio cholera, S for salmonella, and H for our helicobacter, that is Helicobacter pylori or Helicobacter, and Y for Yersinia pestis. Okay, and in here only you can write one more point, that was of our spore forming, SPORE, spore forming. What are our spore forming bacteria? They are Clostridium, that are very commonly asked, Clostridium, and anthrax, ANTHRAX, anthrax. Okay, and while doing questions, if you find more, you can add them also, but they are main. All right, I think everything is clear. We will continue our video in next part. That's all for this video. Thank you so much. And if you have any doubt, comment below. Do not hesitate to ask.